get started? Today we have a lecture on action potential for the cardiac cells as well as the ECG lab. So think of this as a microscopic anatomy. We're going to look at the cells. So to give you an idea, the cell is mostly muscle cells. A very small percent are those autorhythmic cells that have the pacemaker potential, like the SA, the AV node, the Purkinje fibers, all that. The cell is very aerobic. There's a lot of mitochondria. But we're going to focus on the action potentials of these two cell types, the contractile cells and the autorhythmic cells. And so we also want to compare the myocardium, cardiac muscle, to skeletal muscle. Now you learned about skeletal muscle in 430. In terms of what cardiac muscle is, what makes it unique are the presence of intercalated discs. Contractile cells of cardiac muscle tissue. Intercalated discs. Structure not seen in skeletal muscle tissue. These discs have two key components shown in this figure. The intercalated discs include the gap junctions and desmosomes. The gap junctions are shown, to it. The connecting proteins of the gap junction have a pore. They're porous. They're right here in the cell, in this horizontal portion. So think of the intercalated disc as having two parts. This kind of horizontal portion between cells and this vertical portion. The gap junctions are in the horizontal part. And if you recall, they link cells together, but in the case of the heart, the gap junctions will allow efficient spread of the depolarization. Signal starts at the SA node and it spreads to every cell of the heart, mostly because of the gap junctions in the intercalated discs. All right, the desmosomes. The desmosomes are in the vertical part of the intercalated discs. They're kind of like a glue for the cardiac muscle. Act as a glue for cardiac muscle. Let's remember that the cardiac muscle, there's no tendons attached to the skeleton. Skeletal muscles attach to the skeleton, and they have a connective tissue system to help pull on the skeleton, but the heart has no such connections. They're just kind of wrapping around the ventricles to squeeze blood out of it. So to kind of keep the bundles of cardiac muscle tissue from splitting apart when it generates tension and contraction of, during systole, it needs the desmosomes to kind of keep it all glued together. Okay. So pretty much know the desmosomes and gap junctions. We can move on. Here's a simplified picture of the intercalated disc. You can, well, before I do, you should notice that when I teach the histology for this unit, it won't be for a while, you can see them on the microscope slide. You can see these striations. You can see these little dark bars there. That would be the intercalated discs. So you have your striations here in the simplified picture. You got a lot of mitochondria. In the vertical part, you have the desmosome, and then the horizontal part, where you would have the gap junction. Okay, so to compare it a little more with skeletal muscle, here's skeletal muscle here, and here's cardiac muscle. They look kind of the same in the microscope, but there are some differences. One difference is you have the gap junctions here. Here is a similarity of cardiac muscle. Skeletal muscle. They both have striations. Okay, I won't even write it. You should know that already. We cover muscle tissue in chapter 4, the histology chapter. But kind of where it gets, may not have been mentioned before, is here. There's a lot of branching of the cardiac muscle cells. Let's say you have a cell, and it branches, and this branches, and then that branches again, and you get all these branchings. And the reason why that's important for the heart muscle is if the signal is going to spread through the whole heart, say you get an action potential here in this cell, maybe it's in the SA node, okay? And you want that signal to spread, and you have all these gap junctions. I mean, it can do so easily. It can spread because they're all branched together. And so it can spread to this last cell because of the branching and because of the gap junctions, okay? And so the branching is important. Now, in skeletal muscle, when you study that, the muscle fibers are kind of more parallel. Pretend these are all muscle fibers, okay? And the thing about it is, put a box around this. Put a box around that. If you want to make these contract and fire an action potential, a nerve needs to innervate it. And you learned all those nerves. So you have a central nervous system, and a peripheral nerve has to like come off of the spinal cord. And it has to enter our box here, right? And it's got to like, you study the neuromuscular junction. That's the only way you can get these down here to fire an action potential and contract. I mean, not so for the heart. It just spreads by itself. There's no nerve. This requires no nervous innervation, okay? That's why the pacemaker cells are so important. So then this kind of leads to my next point. We say that the cardiac muscle acts as a functional syncytium. Let me write that term down. A syncytium is a fused tissue where the cells, there's no cell boundaries. It's one big giant blob of cytoplasm, just like the syncytial trophoblast that digests in the endometrium. That is a true syncytium in the female reproductive system during development. This is not. The heart muscle cells don't all fuse together. Each cell maintains its cell boundary, but it functions like a syncytium because of the gap junctions. The signal can spread everywhere like it were one cell, but it's not. It's many cells. It just functions like a syncytium. So that term is very important to understand. It's how the whole heart can beat. You know, every time your heart beats in each cardiac cycle, it's because of this concept. So I like to mention that. Then the thing about it is, it's like every time your heart beats, every cell, every contractile cell is contracting. However, in your muscles, it's the all or none principle. It responds to force. If you're moving a light load, you recruit a certain amount of motor units. Let's say this is a motor unit, but it's not enough force because you're lifting a heavier weight. Well, then you have to like recruit another one. If you want to generate more force, recruit both motor units. And the all or none principle is all or none principle is you either recruit a motor unit or you don't. It's on or off. There's no in between. So this red one, either it's on or off, and this blue one, it's on or off. Now you can recruit both of them, or one, but not like 1.5, okay? So if you recruit the red one, how many muscle fibers are you getting? You're getting all 3. It's not like you can do it and only get 2. If the nerve fires, the whole thing, get the whole thing, okay? Recruit the motor unit. Or not. In the case of the heart, if you need more contractile force, every individual cell has to contract harder. They call it the contractility of the heart. We'll talk about that more later. Well, in both cases, you need calcium because the striations, the actin-myosin overlap. If you need to look up cross-bridge cycling again and how calcium plays a role in that, go ahead and do it. Calcium binds the troponin and the tropomyosin. It moves it, exposing the myosin binding site so you can get the cross-bridge formation. Now, if what I just said is like completely foreign to you, you need to review it. We still expect you to know how muscle works. Today I'll focus on the action potentials of 2 different kinds of cells in the heart. Okay, and we will look at skeletal muscle again. So for cardiac muscle, note these two cell types. Just the regular muscle cell shown here on the right, we call it a contractile cell mostly. It's most of the heart. Okay, it's a large majority of the heart. It's the muscle part of the heart, the contractile cells. But then those very small little nodes here and there that spread the depolarization that we talked about last time, the SA, the AV node, the cells in those nodes are called autorhythmic cells. They have the pacemaker potential. So these cells here, cells of primarily the SA and the AV node versus cells of the myocardium. And so what's important is these cells have the pacemaker potential to establish your heart rate, we mentioned that. These cells are the pumping, right, during systole. So they both have their roles. They both need each other. You need the autorhythmic cells to spread the signal throughout the whole heart so that the heart will pump. And so what we do is, well, let's kind of look at, what we do in this chapter is we look at the action potential. That's the basic action potential waveform that you're seeing on that picture there for the autorhythmic cell. For the contractile cell, the action potential looks a little different. It's more like this general shape. And I'll describe it as good as I can in both situations. So for the autorhythmic cell, that's where the signal starts. And we talked about this figure the last time. I walked you through all the different parts of the conduction system, from SA node all the way up to the Purkinje fibers at the end. What we see on this figure here is how much time it takes. If the SA node starts the heartbeat, right there, call that time 0. It takes about, around 200 milliseconds for the signal to spread all the way to the ventricular muscle. So you get the SA node, the autorhythmic cell fires, that makes the atrial muscle fire. When you get to the AV node, there's a bit of a delay, right, about 100 milliseconds, and then the ventricular muscle fires. So that's what they're showing you there. So I want to talk a lot about action potential, membrane potential, and I think it's good to always review the neurophys behind membrane potential, since it might have been a while ago since you looked at that. These terms, depolarization and repolarization, so let's kind of recall what they mean. In physiology, there are cells that are excitable. You can excite them. They can make action potentials and signals can spread and muscles can contract. Some cells are not excitable. The cells that are have this membrane potential. Okay? Let's remember what that is. Well, let's say you have a cell membrane. In excitable cells, you can measure this voltage difference across the cell membrane. Pretend that's a double-layered cell membrane. But let's pretend we're just talking about a cell that's not polarized, that's not excitable. If you were to measure the charge difference on either side, plus, minus, you know, there are ions in the cell that have negativity, positivity. Let's say this is the inside and this is the outside. That situation. Negative, positive, inside and outside. It's like the positive and negative, they're kind of attracted to each other across this resistor, well, cell membrane, right? And it's as positive or negative inside as outside. So if you had your electrodes, you're trying to measure the difference, you know, between the outside and the inside, you're measuring voltage, right? That's what voltage is. It's a difference in charge between two locations. In this case, across the cell membrane. And you'd get 0. You wouldn't measure a difference. So in the voltmeter, this is hooked up to an amplifier, right? You know, the arrow would read more or less 0, okay? There's no polarity, okay? 0, that's pretty easy to understand. Now, what we learned about excitable cells, like when we studied the neuron, you remember the number for resting membrane potential? -70. Remember that? Yeah. Some people do. That means the inside of the cell is negative with respect to the outside. So maybe it's like negative, 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 something like that, right? It's like positive, 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 positive. And there's the potential difference there, so like, you measure that, kind of go through this. It's like, you'd measure that difference. And your voltmeter would like register some negative number. Maybe -70, something like that, right? So that's like polarized. So then we study like, what if you depolarize it? That means you let the positive charge in, like through sodium, voltage-gated sodium channels. So you have like, you have some channel, okay, in the membrane. It's still negative in here. But now you have a channel that allows positive charge in. So the positive enters. And it makes the positivity makes the inside of the cell positive with respect to the outside. So then you call that depolarized, right? And these travel on the inside of the cell. And so if you're measuring that, I'll just kind of draw it here. What's my voltmeter going to read? Negative or positive? It's going to read positive. Okay, so that's depolarized. So then what's repolarized? Well, let's pause here. Okay, let's pretend that this is sodium, that the charge is carried by sodium. So I ask a question here. Compare the permeability of, sodium and potassium during a depolarization. Permeability means those channels are open. So let's pretend these are voltage-gated sodium channels, and you're letting all these positive charges in. What we would say is the permeability of the cell to sodium is much greater than the permeability of the cell to potassium at this time. Okay, and if you're reading it, and it's like, let me draw it over here. Draw something there. Here's 0. Here's -70 millivolts versus time. If you're polarized at resting membrane potential, it's like you're like humming along at -70. And all of a sudden you're depolarized. You go like that, right? And so what you would say is for that side, the permeability to sodium is greater than the permeability of potassium. And let's say that you have these voltage-gated potassium channels. I should use a different color. How about green? And potassium carries a positive charge, and the gradient is such that when these open, potassium leaves. So now you have all this positive charge, this potassium efflux. It's leaving the cell. Okay? So you're repolarizing, right? You're becoming polarized again by letting the positive potassium out. So this repolarizes the cell. So during repolarization, what you would say is the permeability of the cell to to sodium is much less than that of potassium, because these close. The sodium channels close. So that would be like, you repolarize back to rest. So I'll just reverse the arrow. You're letting positive in, letting positive out. That's it. That's depolarization and repolarization. Just think in terms of what the cell is permeable to. So are there any questions on my review of depolarization and repolarization? I wanted to mention that because we talk about that today a lot. So when you have a discussion of action potentials for the heart muscle, they have measured 5 distinctive shapes of action potential at different locations. The SA node, the atrial muscle, the AV node, the Purkinje fibers, and the ventricular muscle. It was cool you did the dissection already. You got to see those, right? Everyone did. You did a good job of that. Well, anyways, if you were to measure the action potential, that's kind of what you would see. We're just going to talk about the first one and the last one. So study this one and that one, basically. What we should do is kind of talk about what is causing the different parts of the action potential in each one. Okay? I kind of have it outlined here. Let's see. Okay, so let's do like some autorhythmic cell over here of the SA node. Autorhythmic cell. And a contractile cell of the myocardium. Then we're going to have on the first point there, sodium. The sodium current is responsible for the depolarization of the action potential in cardiac muscle cells. Okay. So let's get our millivolts. Millivolts. Okay. This is going to be like negative 90. I'll just tell you that. And it's like that's kind of resting membrane potential. Do you remember how you studied, like, there's these graded potentials, like, can you kind of reach threshold and get an action potential? In these kinds of cells, there's no graded potentials. You go straight from rest to depolarization, straight shot, because the gap junctions, you don't need that graded potential stuff. You just get it because of the gap junctions. So, no graded potentials. Go straight from rest to depolarization, and it's carried by a sodium current. So I'll just draw a straight arrow going up, call that 0. Maybe it goes up to something like plus 30. So I'll use my black color. Let's say that's sodium. Okay. That's what we got. Sodium carries the depolarization current for ventricular muscle, not that cell. Okay, let's read number 2. Calcium current is responsible for depolarization in the SA and AV nodes. It also triggers muscle contraction. It delays repolarization in all cardiac muscle cells. So there's a couple of things here. Number 1, calcium current is the depolarization of the autorhythmic cells. Okay, so let me use red here. So I'll use red for calcium ions, and it carries the depolarization here. So there is a threshold potential for the autorhythmic cells. Can't remember the number. I have to cheat and look at the slide. Let me fast forward a little bit. I think it's -40. Yeah, -40. Okay. Oh, whoops. So that -40 is called a threshold potential for this cell. So I'll use red. So a depolarization would just kind of look like this. It just goes up, right? But it's not carried by sodium, it's carried by calcium. So that's one thing that calcium does in the heart for the autorhythmic cells. The other thing it said was there's a delay in repolarization in the cardiac muscle cells. That's the contractile cell. So what happens in the heart muscle, these cells over here, is the sodium channel is open, but they close, they inactivate very quickly. And you start to see that on the trace. It starts to go down a little bit. It starts to repolarize. But it kind of stops. Well, anyways, you should know, when you see this little downtick, that indicates the sodium channel inactivation. It starts to repolarize because of that sodium channel inactivation. But it doesn't continue to repolarize. In the heart muscle, I'll explain why later, you want to delay that. You want to prolong the action potential. So there's calcium currents that prolong this depolarization. It kind of looks like this. Due to calcium, I'll explain why in more detail in a minute. It's just an overview. Boom, that's what it looks like. You're not repolarized yet. You're starting to, you delay it. That's calcium. Okay, moving on, number 3. Potassium current is responsible for repolarization in both cells, the cardiac muscle and the SAAV node. I use green. It's my little key here. Potassium. Okay, so you got repolarization here. This is going to go down to like -50 or -60, something like that. That'll be kind of like another point right here. Let's say -50, depending on what book you read. So anyways, that's the action potential. Okay, just the dipole and then the repole. So for this, the repolarization is carried by a potassium current. Let me see if you're with me here. Is potassium entering the cell or leaving the cell to repolarize in both cases? It's leaving. Very good. Repolarize, back to rest, and boom, you're at rest again. Okay, so that's kind of the whole thing for that one. All right, so the last one is the pacemaker current for the pacemaker potential. They're called funny currents, actually. It's responsible for the pacemaker activity in the SA and AV nodes. They kind of gave it that name, I guess, when they're studying these cells. They saw these currents that just never went away after an action potential. Okay, so let's distinguish here. When I say action potential, for this cell, it's like that whole thing. You got the resting potential, you got the action potential. Now for this cell, I just described the action potential. This dipole and this repole. But what they observed is the funny currents, they'll depolarize these cells. It's a slow depolarization to threshold every time. And so most books will say that that funny current, I mean, there's no element on the periodic table called funny, right? But it's carried by sodium. So it's like you start at -50, and you get this slow depolarization to threshold. Wait, it's carried by sodium, so that's the wrong color, I'm sorry. I use black, so just, boom. Okay, and then you get your action potential, and once you get to -50, the quote-unquote rest, is not a resting potential. These cells, they don't rest. They slowly depolarize the threshold to give you another action potential. And that establishes your heart rate, the time between action potentials. So, yeah, so you call this, the black one here, as the pacemaker potential. Now there's some details I left off for the contractile cell. I want to talk about that first. Any questions on the currents carried by these things? We have sodium, we have potassium, we have calcium too. So that's kind of like a third one that we don't mention in 430. It's very important for the heart muscle. Okay, so let's look at this figure here. This figure, we're not talking about the autorhythmic cells. We'll kind of table that for now. I want to talk about this cell here. One thing you might notice is it's a double graph on the picture here. Here's the action potential that I just outlined. But do you see how they have this other curve here on this y-axis? It's a double y-axis, millivolts tension, because they're also reminding us that this generates force, and so they have a tension curve. So if I could draw that on here, I'll just kind of add to the picture. I'll just make another y-axis, call it tension, and put like a graph there, like that, something like that. That's always what's shown to students, that there's a tension curve. Now these down here match with this. These are the currents. And so they call it permeability, what the cell is permeable to, whether it be sodium, calcium, or potassium. And I made the colors match. I used red for calcium and green for potassium, but I didn't use yellow because you can't really use yellow on a whiteboard, you can't see it, right? So I used black. So that's what I want to talk about. Notice how I put the numbers, 0, 1, 2, 3, for like the different phases of the action potentials. Let's talk about that. So I guess where I have the 0, I put it during resting membrane potential, 0. Put a 0 there. So at 0, my 0, that is when the cell is in resting membrane potential, -90. 0 is rest. A -90 millivoltage, something like that. The cell, the contractile cell, that's what we're talking about here, is not firing. Now, I want you to, as a student of physiology, to basically look at this. This, on the bottom, explains the top. okay? So let's understand that. Look where the 0 is right there. Look at the yellow and look at the red. For the sodium and then for the calcium, the cell is not permeable to it. So they're not doing anything. So look at what the cell is most permeable to. What's the green one? Potassium. When potassium channels allow potassium to leave the cell, that makes the inside of the cell negative. So that explains it, right? You have potassium efflux, the inside of the cell is negative. All right, now we go straight from rest to phase 1. Right here, I put the 1 right next to the depolarization. You basically open the voltage-gated sodium channels. I'll just say E-gated sodium channels. Now, if you look at the yellow curve, it's off the chart. The sodium channels, they're the most plentiful, concentrated channel in the tissue. They're open really fast, but then they close really fast. Okay, they open so fast that the rush overshoots equilibrium. It should just fall to 0, but it kind of overshoots it. Okay, it's called the overshoot. You depolarize to plus 20, plus 30 about, okay? You overshoot equilibrium. Plus 30 millivolts about. Okay, so that's basically depolarization. I think that's all I have to say. And I also said that even though these open really fast, they inactivate really fast. So right after this, sodium channel inactivation occurs, and I left it on the board here, right? It's that little downtick. On the blue graph here, is that little part where it starts to go down. You start to repolarize, but then you stop immediately because the repolarization is delayed. So I put the number 2 kind of where this is happening here. They call it the plateau part of the action potential. I'll put the 2 right around here. Kind of where I had the red line there. I call that the plateau. Repolarization is delayed. You extend depolarization. Different way of saying the same thing. So let's pretend that we have, it helps to draw a picture, at least for me to teach it. Let's pretend we have a sodium channel here and a calcium channel here. I'll use red for calcium like I did before. T-tubule. And maybe you have, I abbreviated SR, the Sarcoplasmic Reticulum inside muscle cells. In the case of the heart muscle, there's a lot of calcium in here. And there's a little gate. These sarcoplasmic reticulum, they're filled with calcium ions. Calcium is very important for muscle contractions. These cells are packed out with them. But in the condition of rest, you don't need it, so you just kind of have it stored in this little stable here called the sarcoplasmic reticulum. So what we have so far is when you depolarize and activate, inactivation is basically you have this ball and chain plugging the sodium channel. What happens in number 2 is shortly thereafter you have voltage-gated calcium channels. If you look at where the number 2 is, you get your peak permeability, this inward flux, during this time. So they call it, your book calls it a calcium surge, I believe. So calcium enters the cell, called the calcium surge. The other thing that happens is this calcium, you want it to open this calcium sensitive channel. So that channel there is calcium sensitive trapdoor, channel, whatever you want to call it. The point is, this surge of calcium, it opens that door, okay, in a way that releases all of this calcium. And that calcium is quite a bit. So I'll just kind of swing the door open a little bit, and then you let all this calcium out. Your book, at one point, I think she wrote it as these calcium sparks. Remember, calcium is for the cross-bridge cycling. So imagine you have muscle, thick and thin filaments overlapping here. Draw a little circle here. The calcium surge isn't enough calcium to get the muscle to contract. So it's like a little something that you need to open this door so that you get the calcium sparks. So you have this remarkable increase in intracellular calcium that allows muscle tension to begin to build. So in terms of events that are happening is, you get this, number 1, this calcium surge, which leads to the release of calcium from the sarcoplasmic reticulum. I'll just abbreviate and call it calcium sparks. And that's enough calcium for the cross-bridge cycling, so tension builds during the plateau phase. And actually, at the end of it, you attain max tension, right here. The tension development during systole, essentially. So tension builds, then you get max tension. And there's one more thing. If you look at the calcium permeability, look at the other 2. During this time, there's a permeability to calcium, but you've plugged potassium. Okay, potassium went down, and so let's call it the potassium plug. You're keeping potassium inside the cell. Maybe I can draw one more channel here. Let's just pretend it's plugged up. potassium, there's a plug there, and it can't leave. So you're keeping positive potassium in the cell. Keeps potassium in the cell. The reason why that's important, it helps extend the depolarization, okay, by plugging it up. So all those things are happening to extend the depolarization. It's actually very important. And now I'm just going to explain the reasons why it is. I'm not going to say why it's important quite yet. So once again, calcium surge, calcium spark. So you get your max tension, and then you plug potassium. And all those four things are happening. And then finally, I put the number 3 there. The calcium permeability drops off, and the tension unwinds, and then the cell becomes most permeable to potassium. Erase all this. Call it repolarization. So the number 3 is right here. You just call it repolarization. Potassium efflux. Allows membrane potential to repolarize, to rest. So I put the key moniker events here, 1 to 6, and I just point to different parts, not all of them, but most of them. If you look at where number 1 is, what I'm going for is, of the list of the 6 things, what caused number 1, the depolarization? It's just one thing. Number 2, the sodium caused it, right? The sodium spikes. What about the plateau, like this number 2 points to? A lot of things. The calcium, the first thing, let's go in order, I guess, while I said it. The calcium surge, then what happened? Then the sparks, you achieved the max tension. You also had the plug. So all of those things occurred during number 2. What's the one thing for number 3 is pointing to? Repolarization. Potassium efflux. Okay, that was it. So I just want to summarize with that. Now the reason why you have this very long action potential, it's like 0.3 seconds, okay? And what you're doing is, by this plateau, you're extending the depolarization. And the one thing I would also add to the 1 and the 2 and the 3 is where I'm pointing with the red arrow. If I say identify what's happening there, you're going to put sodium channel inactivation. Okay, that's the other thing I want to add. But let's remember that if you have a long action potential, you're going to have a long refractory period. Okay, refractory period is a period of insensitivity, and in heart cells, it's important that you have a long refractory period because then you cannot re-stimulate the heart. And that'll allow the heart muscle time to relax, as we'll see. So that kind of leads to my next point here. If you compare cardiac to skeletal muscle, the two muscles will behave in very different ways. When you study the cardiac muscle, we looked at pictures like this. Let's look at the first graph on the left. One twitch. And I have what you should pay attention to, the membrane potential, the tension curve, and the refractory period. For these data slides, the action potential is really quick in skeletal muscle. Okay? And the blue line is the tension curve. Okay? And look at the time here. From time 0, the action potential is just the first 10 milliseconds. But the twitch is about 100. Okay? And the muscle twitch is not how muscle works. I want to remind you of that. The muscle twitch is a lab phenomenon. You know, if you're a physiologist and you're studying muscle in the 50s, because we don't know how it works yet, you want to see how the muscle responds to a single stimulus. That's the twitch. That's what we study in classes like this. And what we see is that when you stimulate the muscle here with the action potential, one twitch is a period of contraction and then relaxation. You say, oh, great, okay. Well then, let's see how muscle responds if I re-stimulate, which you can do, because as long as you're after the refractory period, you can re-stimulate. So the physiologist will just set their amplifier to 75. Let's stimulate every 75 milliseconds before the muscle has completely relaxed, and let's see what it does. And that's the next graph. You kind of do these experiments and simulations, right, where every black arrow you're able to re-stimulate. And what happens is you generate more force, right? This herky-jerky wave summation. Until the muscle's warmed up enough, you get this nice, smooth tetany. Okay? So, let's kind of like take an inventory of all that stuff I just said and just kind of compare. Skeletal and cardiac. So for these two data slides, I guess to make it clear, if it's not clear where I'm going yet, write down how long the action potential takes. Action potential duration. Call it AP duration. Okay, for skeletal muscle, we did that first. It's very quick. There's no plateau phase. It's super quick action potential. It's only about 10 milliseconds. And then note how long the refractory period is. About the same amount of time. The refractory period, this period of insensitivity, is as long as the action potential. So you could re-stimulate any time after that 10 milliseconds because the contraction duration is quite long by comparison. The contraction duration of the muscle twitch. It was about 100 milliseconds. And then so what we saw on the frame on the right, when you re-stimulate, set the amplifier to re-stimulate at a frequency of 75 milliseconds, you kind of like achieve tetany. Tetany is that how muscles work, that nice smooth force generated, the isotonic force there. So let's just ask it as a question, is tetany possible? I present it as a yes-no question, is it yes or no? Is it possible in this kind of muscle to achieve tetany? Yeah, please say yes. Hey, you took 430, you passed. That was a big part of the muscle unit. That's how muscle normally works. You know, the twitching, if your muscles twitch, you look at it like, why are you twitching? There's something wrong. This is how muscles work. They generate smooth force. So that's the whole thing about muscle. Now, if we look at the same kind of data for cardiac muscle, it's quite different. So there's skeletal muscle. Here's the cardiac muscle data. Let's look at that first slide there. For cardiac muscle tissue, these are the contractile cells, right? Not the autolytic cells. Well, as we just seen, you got this huge, long plateau. It extends the length of the action potential to about 250. So the refractory period is that long, 250. If you look at the contraction duration, it's about as long, maybe a little longer. I don't know, we'll call it 260, I guess, about 250. So here's the thing. If you try to re-stimulate every 75 milliseconds, can you? You cannot. You're in the refractory period. And anywhere in here, you try to re-stimulate, you can't. So let's kind of look at that data slide, the next one. So it's like time 0, boom, stimulate. And then you try to re-stimulate, they gray out the stimuli arrows because you can't do it. The first place you can is about 250, right? And so, does that look like tetany, wave summation tetany? No. So the answer is no. So, all this just to say, no, okay, why are you telling me just it doesn't happen? Think about the cardiac cycle. Okay, there's systole, and what's the other thing? Diastole. To ensure that the heart has a period of diastole, filling time, you cannot have tetany. I mean, what that would look like in the heart, well, in the heart, if it was in systole the whole time, you know, it's like you can't fill it. You're just squeezing it the whole time, right? So this ensures diastole, okay? So the logic is, by having a longer AP duration, that's longer, you're gonna have, by definition, you're gonna have a longer refractory period, okay? By having a longer refractory period, this extended period of insensitivity, you cannot re-stimulate the heart muscle. This allows time for the ventricles to relax and fill with blood. Cannot re-stimulate cardiac muscle. Okay. This ensures that the ventricular muscle has time to relax and fill with blood. That's diastole. That's really the point of going through all this, what I just wrote. To have students understand why there always is a diastole, systole, diastole, systole with every cardiac cycle. That pretty much ensures it. So pretty much my next slide, I answered this question with what I just wrote. How does a longer absolute refractory period prevent tetany? That kind of, that chain of thinking answers the question. So if you look at data slides like this, which data set, A or B, is cardiac muscle? I think just by a quick glance, it's easy to tell, right? Which one do you think? B. Yeah. This one we might have to look at a little longer. The numbers are a little different. It's hard to read there. Approximate how long the absolute refractory period is each for the cardiac and skeletal muscle. So based on these basic numbers, 10 and 200, which do you think is correct? Long for cardiac, short for skeletal. D looks good to me. Okay. Last question here. Which muscle type, cardiac or skeletal, has a longer absolute refractory period? Cardiac, and that was the main point there. Okay, autorhythmic cells are next, but I want to make sure you guys have enough time for lab. So before we take our usual break, let me kind of intro the lab that you're going to do today. See all these black cases out there? Have you seen these black cases before? Some of you may be. They're new. We keep our biopacks in. So in there is a biopack unit. I have one here. It's the blue box. And a laptop computer with the biopack stuff on it. Biopack software. Let's see here. So let me show you my messy desktop. When you power up your computer, look for that this little green biopack icon. Double click that. Before you do, I'm going to turn on my biopack power unit.